All right, hey everybody. So I'm moving on to the next part of this whole DNA RNA activity. So I still have our original strand of DNA right here that had that code that I gave to those students. What we did last time or yesterday was we translated this DNA information into RNA information. Actually, when we look at this, it would have been flipped like this to get that information. We're changing our thymines to uracil, so we had to add the coloring on here. Is hard with this green background, I think, but we had to add those white marshmallows on there because that's uracil, and RNA and protein language uses uracil and not thymine. So eventually, this is this code, but in RNA language. And so the next step in making a protein, we had transcription, getting it on this mRNA strand. And then we're taking that information and we're going to make a polypeptide from it. So first of all, we need to read this in codon sequences. Codon sequences are 3 nitrogen bases. So if we look at this first one, actually I don't need that anymore. If we look at this first one, we have orange, so that's A. This one's white, and that one is green. So we have AUG, and this is really important, and this talks about this on your worksheet. It says, note that the start codons for an amino acid, MET, oh shoot, where is it at? It says something about, yeah, so it's MET. So if you look at MET on your sheet of paper right here, MET translates, the code is AUG. So actually the start code on this right here, AUG, this is telling the ribosome, or that rRNA that's reading this and it's going through here, this is where we're gonna start, and then this is where we're starting to build this polypeptide chain, or these amino acids. Okay, so this first one is AUG. That's the first 3, and that codes, as we can see on your sheet of paper on here, AUG codes for MET. Most of the ones that are going to be on your sheet are in this nice little chart right here. If not, there's one on the back that this is different than the one on our notes, but it works exactly the same. Is that you take the first base of the codon, A, you take the second base of the codon, U, and then you take the third base of that codon and you go AUG, and you made it all together to AUG right here, which we already know is MET because that's on here. So the next thing that you would want to do is that you would want to go to the next 3. So we have AUG being the first one. Those are done. Now we go to the next 3. That's our next codon. We have GGC. So now you need to figure out which amino acid GGC codes for. Then this would move down the ribosome, and it would go to this next group of 3, which is CUG. So you need to find what CUG stands for, the next codon. And then the tRNA would bring that amino acid to tied to that polypeptide chain. And we get to this last 3, which is UAG. And you need to see what UAG stands for on that on that codon chart. So that is your codons that you need to read, and I'll do it again just in case you missed it the first time. So we have our whole strand here, which hopefully you have written out under what your mRNA sequence is. When you break that into codons or 3s, you have AUG for the first codon. You have GGC for the second codon. You have CUG for the third codon, and you have UAG for the last codon. You should be able to answer the rest of those questions on that activity and turn that in after this. All right, thanks, bye.